Students, today we are going to start our practical, and the practical which we are taking today is detection and determination of ascorbic acid from the given biological sample iodometrically. So let's see what are the apparatus needed for this practical. So the apparatus needed are dropper, conical flask, burette and beaker. After that we'll see the chemicals which are required. So chemicals required are 0.1 normal iodine solution. After that we'll need 0.1 normal hyposodium thiosulfate. After that we'll need 0.1 normal CuSO4 solution, and lastly we'll need 10% of starch solution. And this is the reaction which we have to perform. So first of all we have got ascorbic acid. After that we have to do this and we'll get the product, that is ascorbate. So before starting the practical, let's see a short introduction about this experiment. So introduction is that the vitamin C, that is also called as ascorbic acid, is essential for human life and is required for a range of physiological functions in human body. It can be found either in fresh fruits and vegetables naturally or in medical forms such as normal tablets and liquids. It is mostly widely taken as a supplement through daily requirements of vitamin C and challengeable according to the age and conditions. It is around 75 to 90 gram per day or for healthy adults, and no more than 2000 mg per day is recommended. It is one of the most essential vitamins ever discovered. Besides, it plays an important role as an oxidant, antioxidant, and free radical scavenger. It has been suggested to be an essential antiviral agent. It is most important in day-to-day life. With this, you can only see that what is vitamin C. It is also called as ascorbic acid, and that is the practical which we will be performing today. And the introduction only tells us that in day-to-day life it's very important, and vitamin C should be used in day-to-day life. Now let's move towards the procedure which we have to do for this. So procedure is, first step we'll see the standardization of hypo solution. For that you will need to prepare 0.1 normal of CuSO4 5H2O solution by dissolving 495 gram of CuSO4 in distilled water in 100 ml of volumetric flask using 5 to 6 drops of sulfuric acid. Means you have to dissolve 2.495 gram of CuSO4 in distilled water, and you have to make up the volume till the mark of the volumetric flask, and in that you will have to add 5 to 6 drops of sulfuric acid. Make up the volume of the mark with distilled water. Pipette out 10 ml of the solution in 150 ml of conical flask. Add NH4OH solution till the solution becomes dark to clean. That the meaning is that when you will do all this procedure, the solution will become dark, but after adding the solution, that is NH4OH, you will get a clear solution. Till that time you will have to do this practical. After that, now add 10 ml of 10% Ki solution and wait for 2 to 3 minutes. Titrate the liberated iodine against hypo solution from the burette till the solutions becomes pale yellow. Now add 1 ml of freshly prepared starch solution with the solution till the solution becomes blue in color. Continue the addition of hypo solution till the solutions become colorless. Note the endpoint. Now when you will do this practical, you will get these types of reading. First of all, when you will do the standardization of hypo solution, first there are 3 columns in that we will see, that is, first is the serial number. After that the volume of CuSO4 solution, we'll name it as V1. After that the volume of hypo solution, and last is the endpoint. So while taking the accurate measurement, first of all we will get 10 ml. Then again we have to take 3 readings of 10 ml. After that you will get the readings in the first one If you have performed, you will get 10.1, second one you will get 10.2, and third one you will get again 10.1. It depends upon you in which way you have to perform, and so the endpoint is 10.1, means V2 will name it as 10.1. After that, the next is the main titration. In the main titration, we will see how to do it is dilute the given solution of vitamin C in 100 ml of volumetric flask and make up the volume to more with distilled water. Means you will have to take the solution of vitamin C in 100 ml of volumetric flask and you will have to make up the volume with distilled water. After that, pipette out 10 ml of the solution in 150 ml of conical flask. Then add 10 ml of 0.1 normol of iodine solution and 10 ml of freshly prepared starch solution. After that, when you will take all these solutions in the conical flask, shake the content for 2 minutes and titrate the unreacted iodine against hypo solution from the burette till the solution becomes colorless. Note the endpoint. And the next readings means the main titration. The readings you will get in the same way, and the readings are 3.4, 3.5 and 3.5, and we have taken the mean of these all three and you have got the endpoint, that is 3.5, and we have written it as the V3, that is the endpoint. After that, the last is the blank titration. In blank titration, how you will do it? Titrate 10 ml of iodine solution against hypo solution from the burette using starch solution as an indicator using the above preparation. The above preparation means the two titrations which you have done in the same procedure, in the same way, you will have to also do the blank titration, but only the difference is that you have to take 10 ml of iodine solution against the hypo solution, and same till the solution will become dark, and you have to titrate it till it becomes colorless, and that is the overall practical which you have to do. And in the third one, the readings which you will get after taking the first one, you will get 4.5, then you will get 4.6, and lastly again you will get 4.6. Again I repeat, it depends upon the way you do the practical. And last you will get the endpoint. Endpoint means when we have taken the mean of 4.5, 4.6 and 4.6, you will get the mean, that is 4.6, and that we have named it as V4. This is the calculation which you will have to do when you will do the practical. After this, let's move towards the last, that is result. Means after calculating all these things, by taking the mean or taking everything, you will get the result, and the result is that 3.522 mg of vitamin C, that is ascorbic acid, is present in 10 ml of the sample of the vitamin C. So with this, I hope you have understood the practical, that what we have done, and in this way you will have to perform the practical. Thank you.